Namaskar, Namaskar. My name is Gyanendra Kumar. I am biology educator in India. In this video, we will discuss counter current mechanism. You all know what is counter current mechanism. This counter current mechanism is that mechanism we that used to explain mechanism of concentration of urine. What mechanism of concentration of urine? What is this mechanism of concentration of urine? Please try to understand. It is the mammal who have ability to concentrate the urine. Mammal, not fishes, not amphibian, not reptiles, not anyone. It is the only mammal who can concentrate the urine. How and why we can concentrate the urine? Because we have loop of Henle. We have nephron, and nephrons have different part. You all know that nephron have glomerulus. Yes, nephron have glomerulus, and then we have Bowman's capsule, and this is proximal convoluted tubule, and then this is descending loop of Henle, then it's ascending loop of Henle, then it's distal convoluted tubule, and then collecting duct present, right? So mammals have functional loop of Henle. This loop of Henle, yeah, this loop of Henle's, which is present in medulla. Yeah, loop of Henle present in medulla, not in cortex of the kidney. Right. So this loop of Henle, and then a network of fine blood capillary surrounding it, that is called vasa recta. They together in mammals, because of these two structures only, the mammal can concentrate the urine. And mammals, they can concentrate their urine even 4 times. Yes, we can concentrate our urine 4 times. Yes, fine. So if urine will be not concentrated, like you imagine a situation where we removed our loop of Henle. So if if our loop of Henle will be removed, then our urine will be highly diluted. Fine. We all know that kidney have two part. When you will take a transverse section of kidney, kidney have two part, right? This outer part is called cortex, and inner part is called medulla. Yeah, this inner part is called medulla. And you all know in the cortex osmolarity, in cortex, whatever interstitial fluid present, that osmolarity is 300 milliosmol per liter. 300 milliosmol per liter, and this osmolarity is almost equal to osmolarity of plasma. But when you will come to medulla, as you will go more and more deep into the medulla, yes, as you will go more and more in deep into the medulla, the osmolarity will increase. Yes, the osmolarity is going to increase, like from 300 milliosmol per liter, it will increase to 600 milliosmol per liter, then it will increase to like 700, 800, 900 milliosmol per liter, and it can reach even 1200 milliosmol per liter, right? Where interstitial fluid of medulla. Fine. And in this same interstitial fluid, of the medulla, our loop of Henle present. Here only collecting duct present, and this loop of Henle is surrounded by one network of blood capillary, that is called vasa recta. Actually, students, try to understand it. See, this is my, if this is ascending loop, this is efferent arteriole. This is efferent arteriole. Okay, and this is efferent arteriole. So, and this is our Bowman's capsule, right? And this is proximal convoluted tubule. Okay, and then we have loop of Henle, hairpin like loop of Henle we have. Yes, we have hairpin like loop of Henle, and then we have distal convoluted tubule. Yes, this is distal convoluted tubule. I'm writing here, this is proximal convoluted tubule. Now a branch of capillaries rise from our efferent arteriole, and it's a form a network around loop of Henle. Yeah, branch from the efferent arteriole will rise and it's form a network like structure around loop of Henle, right? That branching is called peritubular capillary, and in the peritubular capillary whatever part it's forming network like structure around loop of Henle, that will be called vasa recta. Yeah, this is vasa recta. Fine. So what is vasa recta? This vasa recta is part of peritubular capillaries. Peritubular capillaries. And what are these peritubular capillaries? They are the blood capillaries which are starting from the efferent arteriole. Fine. Okay. So this vasa recta and loop of Henle participate in concentration of urine, and this all either it's a vasa recta or loop of Henle, that everything is arranged in this region, medulla region, right? Medulla osmolarity is high. Why osmolarity is high? Because in this medulla, lot of urea and NaCl is retained in interstitial fluid. The fluid present between the cells, fluid present between the tissues, urea is retained. If you remember, students, hello, if you remember, it is the collecting duct who retain a little urea, isn't it or not? And that urea come to interstitial fluid. So that urea plus NaCl it make the concentration in this region high. What I am doing, I am taking loop of Henle here, I am taking vasa recta here, and I am just zooming it, right? Like this structure actually, I am just zooming it. And for my convenience, I am taking this vasa recta separately, and this for clarity, more clarity, and this ascending and descending loop of Henle, I am taking it separately. Fine. This counter actually, now I am going to explain how high concentration means, I mean how gradual high concentration is maintained in medulla, because this gradual high concentration only is going to decide how our filter rate is going to concentrate, how our urine is going to concentrate. Fine. Okay, the whole story start from collecting duct. This is in this diagram, this is our collecting duct, this portion is our actually renal medulla, and this upper portion where this, I mean this all region is actually it's a cortex, this region is medulla. So we will talk the things in medulla because it is the medulla where concentration of urine occurs. So see here, so our collecting duct retained some amount of urea. Fine. That urea it will be absorbed, and then it will come to interstitial fluid. Fine. And from interstitial fluid it will go to ascending loop of Henle. Yeah, it will come to ascending loop of Henle. I am writing here, this guy is our ascending loop of Henle. Fine. So it will come to ascending loop of Henle, and then from ascending loop of Henle, that urea come to descending loop of Henle, and from descending loop of Henle again that urea reabsorb. Fine. And it's go to ascending loop of Henle. Now why they are playing like this? Why they are playing with urea like this? From one side they are reabsorbing and again they are bringing here. Actually result of result of movement of the urea will be what, high level of urea will be maintained in this whole region. Yeah, urea will diffuse and high level of urea maintained in interstitial fluid. So for concentration of urine, first we have to maintain higher osmolarity into the interstitial fluid, and first factor is our urea. Fine. And then you all know our ascending loop of Henle. Ascending loop of the Henle is the great place for absorption of NaCl. and this NaCl absorbed by active transport also, and I mean the and other minerals also actually they absorb. Fine. So lot of phenomena occurs. I'm not going there, right? But here you see ascending loop of Henle, they absorb NaCl, and that absorb NaCl come to interstitial fluid, right? Water will never be absorbed here. Fine. Water absorption will be minimum in this region. Fine. So water will never absorb, but NaCl absorb. Now you tell me, if this NaCl absorb, it will diffuse here or not? It will diffuse into the medulla or not? Yeah, it will diffuse into the medulla, and then it will flow, and then it will come to descending loop of vasa recta, descending limb of vasa recta. Hello, vasa recta, what they do, they collect blood from this interstitial region of medulla and take it to efferent arteriole. Yeah, they take it to efferent arteriole like this. So you see here blood is flowing down here. In vasa recta, blood flow. Blood is flowing down, right? So we will call it descending vasa recta. Please do not confuse with descending loop of Henle. This is descending vasa recta. Okay. So actually that NaCl, it will enter in this vasa recta, descending vasa recta. Fine. Its flow, it's absorbed here, then it entered into descending vasa recta, and then that NaCl went to ascending vasa recta, and then that NaCl reabsorbed again in interstitial fluid. Fine. From ascending loop of Henle, the NaCl was absorbed in interstitial fluid. Then it was secreted in descending vasa recta. It entered in with descending vasa recta. Okay, and then it went to ascending vasa recta, and then again it secreted to interstitial fluid. What will be result of this second phenomena? Result of this second phenomena is that high concentration of NaCl will be maintained in this whole region. It is the two things, urea and NaCl, maintained high concentration in this region, and result of that urine is to concentrate. Fine. This is what our counter current mechanism. Any question? If you have, please ask. Now how question can be asked? Try to understand in exam. What are the solutes who maintain high osmolarity in this region? So first one is NaCl and second one is urea. NaCl and urea. Okay. Ascending limb of loop of Henle or descending loop of Henle? Which one absorb NaCl? So it's ascending loop of Henle absorb NaCl. Fine. And okay, water who can absorb? So it is the descending loop of Henle who can absorb water. In which part of the nephron absorption will be minimum? So it is the ascending loop of Henle where absorption will be the minimum. Okay. Which part of the tubules of the nephron absorb urea? It's a collecting duct absorb a little bit of the urea. What will be result when this urine will not be concentrated? If urine will not be concentrated, our urine will be multiple times diluted. This is what our counter current mechanism. And because you see here filter rate is flowing in one direction, blood is flowing in opposite direction, that's why we'll call this whole mechanism counter current mechanism. Remaining we'll see in next video. Bye.